Welcome back, friends. So in my today's video I am going to discuss on the development of the female gametophyte as well as the male gametophyte in Cycas plant. So here in one of my video I had already discussed on the development of the megasporophyll, the structure of the megasporophyll and the development of the megasporangium, that is the ovule, and also how the megaspore mother cell will divide by meiosis to give rise to a linear tetrad of 4 megaspores. Okay, this is a linear tetrad of 4 megaspores. So the structure of the ovule I had already discussed in one of my videos. So in today's video I am going to discuss on the development of the megaspore. And okay, let's get started. So here, if we talk about the megaspore, as I had mentioned earlier that the functional megaspore is the first cell of gametophytic generation, and it remains embedded inside the nucellus, which will develop to the female, which will give rise to the female gametophyte. Okay, so out of these linear tetrad of 4 megaspores, out of these spores we know that the lowermost one, the megaspore, that is the megaspore which is facing towards this chalazal end, will be the functional megaspore. Okay, it will become the functional megaspore, while the rest 3 megaspores will degenerate. So you can see here in this figure that the 3 megaspores, they degenerate, whereas the one which is facing the chalazal end will become as a functional megaspore. So here let us take a look at how this functional megaspore will develop inside the nucellus. So here, first thing, what will happen? Now this functional megaspore, that is the female gametophyte, is, we call it monosporic. Why? Because out of the 4 megaspores, only one becomes functional. So the female gametophyte is monosporic. Okay. Now the development of the female gametophyte, it takes place entirely within the microsporangium or the ovule. So just we will take a look at the development of this functional megaspores. Now this functional megaspore, each of the functional megaspore is regarded as the first cell of the female gametophyte, and it remains enclosed within the ovule, inside the ovule, and also it will germinate in situ. Okay, it will germinate in situ, meaning in its original place inside the nucellus of the ovule. So what happened to this functional megaspore? Prior to germination, this functional megaspore enlarges in size. Okay, it enlarged in size by absorbing some of its neighboring cells. Okay, the neighboring cells which is there in the nucellus will be absorbed by the functional megaspore, and thus the functional megaspore will enlarge in size. And also what happened, this functional megaspore is surrounded by a tough membrane, and also its nucleus is dividing. Okay, so it absorbs the cell, the It absorbs the neighboring cell, becomes enlarged in size, it is surrounded by a tough membrane, and also the nucleus divides by free nuclear somatic division, forming numerous free nuclei. We can see here. It starts to divide and divide, forming many free nuclei, forming numerous free nuclei. Okay, now these free nuclei which are formed, they are distributed in the cytoplasm. But what happened later on, a big central vacuole, as shown in figure E, a big central vacuole appears. Okay, it appears in the middle, which thus pushes these free nuclei to the peripheral protoplasm. So these, due to this big central vacuole which appear, this big central vacuole, it uh, causes the free nuclei to be evenly distributed. It pushes these free nuclei to the periphery. So what is the next step? Now these free nuclei are mostly distributed in the peripheral layer of the cytoplasm surrounding a big central vacuole. So what happened next is that a cell wall formation between these free nuclei will take place. How it will take place from the periphery, we can see here from the periphery extending towards the center until the megaspore vacuole is filled up with a cellular tissue. You can see here in figure I, whereby due to the formation of the cell wall which extend from the periphery towards the center, now even the megaspore vacuole is filled up by a cellular tissue. As a result, a solid mass of the gametophytic tissue called the endosperm tissue is formed. Okay, this is the endosperm tissue is formed, and it is also haploid. Okay, this endosperm tissue is haploid. Now, while the megaspore increases in size, its germination starts. And the surrounding nucellar tissue, we know that this one is the nucellar tissue. The surrounding nucellar tissue, it forms a layer around the germinating megaspore. Okay, so the nucellar tissue, it forms a layer. Okay, it forms a layer around this megaspore, and this layer is referred to as the endosperm jacket. Okay, so this cellular tissue, cellular female gametophyte, is referred to as the endosperm, and the layer which surrounded a nucellar tissue will form a layer which will surround this endosperm, and that layer is known as the endosperm jacket, and it is also nutritive in function. Okay, it is nutritive in function. Now you can see that this layer, it closely resembles to the tapetum of the other plants. Okay, what is the tapetum? It is the specialized layer of nutritive cells. So this endosperm jacket also, it acts as, it serves the function of nutrition. It provides nourishment to the developing embryo, endosperm. Now the gametophytic tissue, it shows two region. Okay. So remember this one, it's very important that the region of larger cells are found near the chalazal end, while the region of smaller cells, they are found towards the micropylar end. And the Archegonia begin to develop on the germinated megaspore. So here remember one thing, that the gametophytic tissue, it shows a smaller cell towards the micropylar end and a larger cells towards the chalazal end, near the chalazal end, and also the archegonia will begin to develop on this germinated megaspore. Okay, we know that the archegonia is the female sex organ. It is the egg producing organ. Okay, and an egg producing organ will start to form in this germinated megaspore. So now let us take a look at the development of the archegonia. So for this one, I'm going to show you this picture. Okay, it is much more clearer. Now, for the development of the archegonia, some superficial cells, okay, some superficial cells around 2 to 6 of the female gametophyte, okay, which lie towards the micropylar end. Okay, so that means here some of the superficial cells which lie towards the micropylar end, those cells will become differentiated into the archegonial initial. Okay, they will become differentiated into archegonial initials. Let us take a look at only one archegonia. How does it form? We know that around 2 to 6 of these cells will become differentiated into archegonial cell. So let's take a look at how this archegonia will form. Now each of the archegonial initial cell, they will divide by periclinal division. Okay, they will start dividing by periclinal division. Now what happens after they start dividing? They will give rise to two unequal cells. You can see here in figure B, it is very clear that these cells are unequal in size. A smaller outer one is referred to as a primary neck cell, whereas the much larger central cell, okay, the much larger one is called the central cell. So the archegonial initial will divide by periclinal division to give rise to a smaller primary neck cell and a larger central cell. Now next, the primary neck cells will again divide, but here it's not a periclinal division, it is anticlinal division. Okay, it will divide by anticlinal division to form two neck cells. You can see here these two, they are the neck cells which form by the division of the primary neck cell. And what happened to the central cell is that the central cell will enlarge in size. It will enlarges in size and becomes vacuolated. Okay, so first it will enlarge in size and becomes vacuolated. Soon after this, it gets surrounded by an archegonial jacket. Now there is the layer which will surround the central cell. That layer is referred to as the archegonial jacket. Again, the function of the archegonial jacket, it acts as a nutritive layer. Okay, again this archegonial jacket, just like the endosperm jacket, it acts as a nutritive layer. Now what happened just before fertilization? The nucleus of the central cell, here the nucleus of this central cell, it will divide by mitosis to form a small ventral canal nucleus. Okay, you can see here first there is a vacuole which appear, and this one is the archegonial jacket which serve the function, which acts as a nutritive layer, and the nucleus will start dividing to give rise to a small ventral canal nucleus and a much larger egg cell, a much larger nucleus. This one, a much larger nucleus of the egg cell. Okay, so it is the egg nucleus. But here there is no wall formation between the two nucleus. They remain as it is. There is no wall formation between the two nuclei. Now the neck canal cells are completely absent. The neck canal cells are completely absent. So here we can see the ventral canal nucleus. It will move towards the egg of the the ventral canal nucleus. It will move towards the neck cell, okay, towards the neck of the archegonium. You can see here, this is the neck cell. So the ventral canal nucleus, it moved towards the neck of the archegonium, and later on it will degenerate. So you can see here in figure D, there is no ventral canal nucleus because it has already degenerated. Okay, it will get degenerated. Now this is what we call the major archegonium. Okay, this one is the major archegonium. Thus the major archegonium, it consists of only two neck cells, a ventral nucleus, and an egg. Okay, this is the egg. Now the growth of cells immediately above the archegonia, they become restricted, whereas the surrounding cells grow more rapidly. Okay, so for that I will show you this one. Okay, so here the growth of the cells immediately above the archegonia, they become restricted, whereas the surrounding cells grow more and more rapidly. Now, by the growing of these cells, it results in the formation of a shallow bowl shape. We can see here. This is a shallow bowl shape cavity above the archegonia. You can see here, this is the archegonia. These are the archegonia, and this is a cavity, a bowl-shaped cavity, which is referred to as the archegonial chamber. Now the neck, these are the neck cells, two neck cells we have already seen in the previous slide, that there are two neck cells. So the neck of the archegonia, it open up in the archegonial chamber, and this archegonial chamber, it is full of liquid. Okay, it is full of liquid during fertilization. So here, if you remember how the young ovule it looks like, you can see here only the tetrads, okay, the megaspore tetrads, where by later on when this functional megaspore will develop. So how the major ovule look like? This is the LS of the major ovule, where we have the archegonium and the egg cell, which will fuse with the pollen grains or the male gametophyte to form a zygote. Okay, so now next we will study, or we'll take a look at the development of the male gametophyte. So one thing here I want to tell you, that the male gametophyte development, we already studied that it developed inside the microsporangium and inside the pollen chamber, that is inside the ovule. So here, okay, this is the development of the male gametophyte. Last time I had discussed on the development of the microsporangium and how each of these microspore mother cells will divide to give rise to the spore tetrads, which will give rise to the formation of microspores. So there are a lot of microspores which are produced inside the microsporangium. Okay, so now we will take a look at the development of each of the microspore. So as I said, that the microspore, it will develop inside the microsporangium before pollination and also after pollination. Okay, after pollination when it reaches the ovule. So the male gametophyte, now the microspore, here we will take a look at a single microspore. The microspore is the first cell of the male gametophyte. Now it is uninucleate and it is also haploid. Okay, a haploid spore which is produced inside the microsporangia of a male cone. Now each spore, when just formed, it has two-layered wall. Okay, you can see here two-layered wall. The outer, which is called the exine, and the inner, which is called the intine. Now the spore is also filled with starch grains. Okay, and these starch grains, they will disappear when this spore matured. Okay, when it matures, it will these starch grains will start to disappear. So the each spore, it also has a longitudinal slit. You can see here it has a longitudinal slit so that it appears like a boat shaped. Okay, you can see here figure B and C. It appears as boat shaped. Now the development of the male gametophyte partly occurs inside the microsporangium and partly inside the ovule. So that is why we will first take a look at the development of the male gametophyte inside the microsporangium. Okay, how it develops inside the microsporangium, that is before pollination, before pollination occurs. So now each microspore, it germinates in situ, that is while still enclosed inside the pollen sac of the microsporangium. Now, first of all, what happened, if you take a look at this figure, first of all, what happened, a big central vacuole appears at the center of the spore. Okay, it appears at the center of the spore, thus pushes the nucleus to go on one side. You see here, the nucleus is in the almost at the center, but here it has been pushed towards one side to go on one side due to the formation of a big central vacuole. Now the microspore, it starts dividing. Okay, it starts dividing when it becomes about 3 weeks old. Okay, when it becomes about 3 weeks old, it will start dividing. Now prior to germination, the vacuole disappears. Okay, you can see here in these figures where the vacuole already disappear, and the nucleus, if you take a look at the nucleus, it is dividing. Okay, it is dividing mitotically to give rise to two daughter nuclei. And also not only that, here in figure G you can see that the vacuole completely disappears. This due to the division of the nuclei to give rise to two daughter nuclei. Also what happened, a cell wall formation. Okay, a cell wall is laid down, this one. The cell wall is laid down between the two daughter nuclei. Now this due to the formation of this cell wall, it will cut down a small lenticular prothelial cell. This small cell is referred to as the prothelial cell towards the proximal end, and a large antheridial cell at the other end. So we can see here it has already divided to give rise to two cells, the prothelial cell and the antheridial cell. Now the prothelial cell, it is persistent. Okay, the prothelial cell, it is persistent and it represents one celled vegetative body of the male gametophyte. It does not divide any further, and it remains completely dependent. So, but what happened to the antheridial cell? The antheridial cell, it will again divide, okay, by transverse division. Now it divides by transverse division, forming a small generative cell. You can see here a small generative cell and a large tube cell. Okay, so the protherial cell does not divide any further, it remains as it is, whereas the antheridial cell will give rise to a small generative cell and a larger tube cell. Now the generative cell, it has a small nucleus and lies in a very close contact with the prothelial cell, whereas the tube cell, on the other hand, it has a large nucleus and occupies a greater area. Okay, so it takes about 1 week to form a wall. This wall, you can see here, this is the wall between the generative cell and the tube cell. So it takes about 1 week to form a wall between the tube cell and the generative cell. Now we can see here that this is a three-celled stage of the microspore. You can see here this is a three-celled stage. Why three-celled stage? Because here we have a prothelial cell, a generative cell, and a tube cell. So we have to remember that it is at this three-celled stage that the microspores or the pollen grains, they are shed from the microsporangia of the male cones and they are pollinated. Okay, so this is the how the male gametophyte, the microspore, developed inside the microsporangium. Now it is at this three-cell stage that the uh, male gametophyte or the microspore will be released from the microsporangia and are pollinated. So first, now before looking at the development of the male gametophyte after pollination, that is inside the ovule, so first let us take a look at pollination. Okay, how the pollination occurs, so that we can study the development of male gametophyte after pollination inside the ovule. Now pollination, the pollen grains, that is the microspores, are liberated from the microsporangia at a three-cell stage. That one I had already mentioned. So in Cycas plant, the pollination is anemophilous. Okay, it is anemophilous, that it is wind pollinated. So that means the pollen grains, they are carried through air. They are carried to the pollen chamber of the ovule by wind. So here I'll just show you this one. This is the pollen chamber. So now the pollen grains will be carried by the will be carried to this chamber of the ovule by wind. So at the time of pollination, how does that happen? At the time of pollination, a mucilaginous liquid drop, okay, a liquid drop, or what we call a pollination drop, oozes out at the micropylar end of the ovule. This is the micropylar end of the ovule. This is the ovule. Okay, so it will a pollination drop oozes out from this micropylar end, and the pollen grain which are floating in the air, they are caught in this drop. You can see here few of the pollen grains, they are caught up in this mucilaginous drop or a pollination drop. Now as the drop dries up, the pollen grains, they are drawn inside, okay, to the pollen chamber. Then due to the further drying up of the pollination drop, the pollen chamber is closed. Okay, so when they are they have been taken out. Sorry, when the when the pollen grains, they are already, when inside the ovule, they are caught up in this pollen chamber, and due to the further drying up of the pollination drop, the pollen chamber is closed. Now it is closed, so the pollen grains remain inside the pollen chamber, and also they lie inert inside the pollen chamber for about 4 months. So it is not as soon as the pollen grains are reached inside this pollen chamber they will start to germinate. In fact, they remained inert in this pollen chamber for about 4 months, before germination. Now, at the time of pollination, the pollen grains are directly deposited on the nucellus. This is the nucellus. We know that this one, you can see here, the nucellus of the female gametophyte. So now the pollen grains are already present here inside the ovule. So they will again start to germinate. Now let us take a look at the development of the male gametophyte after pollination. That is, inside the ovule. So now further development will take place. This one. Further development of the male gametophyte takes place after pollination. So we know that the pollen grain remain inert inside the pollen chamber of the ovule for about 4 months. After that, they start germinating again. So what happened? Let us take a look at the different steps of germination of the microspore. Now we know that the microspore, it is in a 3-celled stage. Remember, if you remember, before pollination, it has already germinated to give rise to the 3-celled stage of the microspore. That is the prothelial cell, generative cell, and the tube cell. Now, here the tube cell, this one, the larger one, that is the tube cell, it will elongate. It elongates, breaking the exine to form a slender, long branched pollen tube. This tube cell actually, it will elongate to form a pollen tube, whereas the generative cell, it will become larger. The generative cell will become larger, and also it starts to